All right. So now let's talk about osmosis. So osmosis, sorry, not about osmosis. Let's talk about hypotonic and hypertonic solutions. Right. So we've been talking about osmosis, and again, that is water going from high concentration to low concentration. Right. So what happens when you put a cell into different types of solutions? So right here you have the word hypotonic solution. Right. If you put a cell in a hypotonic solution, you're putting it in a solution that has less solute. Right. So as you can see right here, the cell has 90% water and 10% solute, whereas the solution right here has 100% water and 0% solute. Right. So if you're thinking about this right here, you got to ask yourself, where will the water go? Remember, water goes from where there's a high concentration of water to where there's a low concentration of water. So what is going to happen to the water here? Water goes from high to low. So let's see. Here you have 90% water. Here you have 100% water. This is a higher percentage of water. This is a lower percentage of the water inside the cell. So the solution has more water. The cell has less water, right? So the solution, let's see what happens when you put a cell into this hypotonic solution. So what's going to happen to the cell? Well, if you put a cell in here and it's again got 90% water inside the cell, but the solution has 100% water, then water is going to go inside of this cell, right? Water is going to rush in here. And then eventually, when enough water gets in there, the cell will burst, right? So, this is why when you get a IV bag at a hospital, that they got to make sure that that IV bag is not 100% water. Because if they connect you to a IV bag that has 100% water, then it will not go well for you because that means that this is what is going to start happening to your cells. Right. So, whenever you have those IV bags that they connect to you in the hospital or to people in the hospital, it usually makes the solution or the solution in there is usually very similar to what you would find in your cells, right? Because you want it to be the same inside and outside of the cell. Because again, this is what can happen if you put your cells into pure water. And so in a hypotonic solution, it contains a lower concentration of solute, right? And that's what we saw right here. There was no solute right here, 0% solute right here. So contains a lower concentration of solute and therefore a higher concentration of water, right? Because it was 0% solute, it was 100% water. And so if a cell is in a solution that is hypotonic to the cell will gain water. And eventually, if it takes in too much water, this will happen right here, right? And this is what you call, let me write this out right here. This is what you call lysis. Change the color. Um, well, change the background in this case. Uh, Anyways, yeah, that works right there, right? So lysis. And if you wanted to be more specific, cell lysis, right? The red blood cell placed in the distilled water eventually lyses or bursts. And so that was a hypotonic solution, right? Hypo. This one is hypertonic. Hypertonic solution, and whoops, I kind of gave away the answer right there. Hypertonic solution is more solute, right? And you see that right here. There is 50% solute right here, only 10% solute right here, right? So it's just pretty much the opposite, right, of what we saw earlier. If you put something in a hypotonic solution, it's going to swell up and eventually it can burst. But if you put this in here, remember, water is going from where there's a high concentration. So there is more here, 90% water compared to here, 50% water. So water is going to go from high concentration inside the cell to low concentration outside of the cell. Right. So this is why you do not want to drink cell water or sorry, salt water, because this is what will happen to your cell. Water is going to leave your cells and eventually your cells will kind of shrivel up the way this cell has right here. Right. So where will the water go? The water will leave the cell and come into this solution right here. Basically, water always wants to try to dilute whatever solutes there are, right? And make it less, make it more water, less solute. Right. So in a hypertonic solution, it contains a higher concentration of solute and therefore a lower concentration of water than the solution to which it is compared. So again, it's basically just saying if you put a cell inside of a hypertonic solution, then it's going to shrivel up, right? This is that red blood cell placed in saltwater solution. Water leaves the cell, and that's why it looks all shriveled up like this, right? Because water was leaving the cell, right? And it was going into that hypertonic solution trying to make it less salty. If a cell is in a hypertonic solution, then water will diffuse out of the cell and into that hypertonic solution, right? So again, y'all, this is osmosis, water going from higher concentration to lower concentration. And so I mentioned earlier that if you go to a hospital and you get an IV bag, and an IV bag are just those things if you're dehydrated or if you need liquids through your body, they will put those and attach those to you. And but you want to make sure that that solution is isotonic, right? You want the same amount of that IV solution as you have in your cells, right? So here 90% water, 90% water, 10% solute, 10% solute. So if you have the same on both sides, this is what you should expect from this right here. Nothing, right? The cell will just go on doing what it's doing because you have the same amount of water. Water will still go in and out of the cell, but it will do it at an equal rate. So the cell's not going to swell up and burst, or it's not going to shrivel up, right? It's just going to stay the way it is, but there will still be a water moving back and forth, just at an equal rate, right? So no swelling and no shriveling up. And this is an isotonic solution. So remember, the hypotonic solution causes the cell to burst because water rushes into the cell. A hypertonic solution causes the cell to shrivel because water rushes out of the cell in a hypertonic solution, right? Think of salt water. And then here in an isotonic solution, it doesn't do anything because it is equal inside of the cell and outside of the cell. Right. So that is what you want if you have an IV bag, because you want the same amount of solutes and water inside of your cell as outside of your cell. Right. So when the concentration is the same inside and outside the cell, you have this isotonic solution. Right. And an isotonic solution is at equilibrium. So there is no net change. Water flows in and out of the cell, but at the same rate. Right. So again, the water will not swell up and burst, or it will not lose water and shrivel up if it is in an isotonic solution. It is at equilibrium. So no net change in the cell. So it's important to remember when you're talking about osmosis that water moves from high to low concentration of water through a membrane, right? That is the definition of osmosis. And that's what we saw in the last few examples, right? In a hypotonic solution, it's going from a high concentration outside of the cell to a low concentration inside of the cell. And it's going to make the cell bigger, bigger, bigger until eventually it can burst. Right. If it is in a hypertonic solution, that means that you have a high concentration inside the cell and a low concentration outside of the cell of water. And so that way, water will leave the cell and shrivel up. Increasing the concentration of the solute in a solution decreases the concentration of water, right? So if you have 0% solute, you have 100% water. But if you have like 70% solute, you only have 30% water. And so water moves from a hypotonic solution to a hypertonic solution until both solutions are isotonic to each other and at equilibrium. So that's what it wants to get to, right? It wants to be able to have the same amount of water inside the cell as outside of the cell, right? And wants to reach this equilibrium right here. Sometimes it can do that, but other times it'll take in too much water and the cell will burst, or other times it'll lose too much water and the cell will shrivel up, right? But eventually, what it wants to get to is this equilibrium. So you have the same amount going in as you do coming out of the cell, right? And so the cell doesn't burst or the cell doesn't shrivel. It just stays that way because it is at equilibrium. All right, so let's get back to the cellular transport. All of these things we talked about are important to understanding cellular transport. Right, so here you see the phospholipid bilayer, right, the plasma membrane. You see the hydrophilic heads and the hydrophobic tails. And then this right here is that protein that's helping these get across. Remember, y'all, sometimes particles that need to get in or out of the cell or molecules are the opposite of this. This guy is hydrophilic, and so usually it attracts hydrophilic things, but again, these are hydrophobic. And so things need to get in and out, but they can't because they're not hydrophilic or hydrophobic. And so they need these proteins to help them get through. right? And this is what I was just saying. Many molecules cannot move through the hydrophobic membrane. And so some molecules must undergo passive transport by traveling across the membrane through a transport protein, right? So here again, you see the word passive. So does passive mean that you need energy or that you do not need energy for this? So yeah, for this one, transport, you're moving across. It does not need energy because it is passive. It is going from high concentration. Here you see this high concentration of these little red particles, and they're going to areas of low concentration. So this does not require energy because it is passive, right? Water moves through special transport proteins called aquaporins. And I mentioned this before a while back, but remember, if something ends in IN, it's most likely a protein, right? And so this black thing right here is that aquaporin, and it's letting water move through there in this case, right? This is called facilitated diffusion and still requires no energy because the molecules are going from high to low concentration. But the reason why, and let me find a different color right here. The reason why this guy is facilitated, if you facilitate something, that means you help something, right? So it is facilitated because it is being helped by this protein right here, this aquaporin, right? So facilitated, though, still does not use energy, and that is why it is considered passive, right? It's still getting some help from this protein, but since it's going from high to low, it is still passive transport. And transport just means movement, y'all. Whenever you hear that word or see that word, it just means to move. Right. So right here, let me shrink this down or bring it down a little bit. So transport proteins, there are two types of transport proteins. There are channel proteins and carrier proteins. And we'll see that right here. So channel proteins are tube-like and provide openings through which particles can diffuse, right? So since they can't get through the parts of the plasma membrane, they need a little tunnel to get through there. And so that's what these channel proteins are, right? They're tube-like or tunnel-like, and they provide those openings which allow things to go from remember, to diffuse means to go from high concentration right here. You see there's a high concentration right here to the low concentration right here. There's only one, two, three, right? So high concentration to low concentration. But again, channel proteins are just like tunnels, right? So carrier proteins, on the other hand, they have a specific shape that fits a specific molecule, right? So when the proper molecule combines with the protein, it changes its shape and it moves across a molecule. It moves a molecule across the membrane, right? So again, it's making contact with one of these molecules that are need to get across. Once it gets there, it opens up. Basically, it changes its shape, brings it across, kind of carries it, right? Carrier carries it across the plasma membrane or the cell membrane, and then it lets it go. Right. This is also going from high to low concentration. So remember, y'all, this is passive. All right. Now we've been talking about passive in the past, right? So now we're going to be talking about active. Kind of just the opposite, right? Passive meant you were at the high point and you moved to the low point. You did not need energy to go from high to low. But in this case, active transport, you're going from low to high. And so you're climbing, you're going against that concentration gradient. And so for this one, like it says right here, it's going to be hard work, right? So this one is going to require energy. Right? Can a cell ever move particles from a region of lesser concentration to a region of greater concentration? Yeah, it has to. If it didn't, we would not be here, right? So this does have to occur, but it requires an input of cellular energy, ATP. Remember, we talked about ATP last time. Whenever you're thinking of ATP, you can think of it as the currency of the cell. If you do anything, you need to pay for it in ATP, right? And in this case, if you're going against a concentration gradient from low to high, you're going to need ATP for that. So here we have an example of a membrane pump. The transport of materials against a concentration gradient from low to high, it's going to go against a normal high to low, right? So that is going to require this energy, which we know is ATP, and is called active transport. So remember, y'all, active transport goes from low to high. Right. And so here you see these are the ADP right here, and this is ATP. So you're having to use this ATP right here to go from an area of high concentration, and you really can't see them right here, but you would remember that this is a low concentration going into the high concentration. So active transport uses ATP, and here we talked about ATP before. So ATP, when it has energy, is known as ATP, but when you get rid of some of that energy, it becomes ADP. Right, but again, y'all, the important takeaway for this is to remember active transport uses ATP because it's going from low concentration to high concentration against the gradient. So if it's going against the gradient, if it's going from low to high, it is active transport because it uses energy. It's like trying to walk against a current in a river. So endocytosis is pretty cool. Some cells can take in large molecules or groups of molecules or even whole cells through endocytosis, right? It requires the input of ATP, but basically endocytosis means it like swallows something up, right? It's like eating something or taking something inside. Exocytosis is just the reverse process of endocytosis, right? It's when cells use this to expel waste and secrete cellular products. So if there's something inside the cell that it does not need anymore, it just releases that waste through exocytosis, right? And here you can see these little things right here are vesicles. And I mentioned earlier, vesicles are like vehicles. They travel, they move things around the cell. In this case, these little blue particles, it wants to get rid of them. So it goes up to the cell membrane, attaches, and then it releases it, right? It expels the waste, it releases it. But this one is important to remember. Remember, it requires an input of ATP, it requires energy. And if it requires energy, it is active transport. Remember, passive, no energy required, but this one is active, energy is required. Right, so here you see this image, and here you see the lipid bilayer, and then there are different types of proteins. Simple diffusion is just something going from high concentration right here. You see this high concentration, the low concentration, right? And so if it's doing that, it does not need energy. So all of these are passive, right? Simple diffusion, channel mediated, and carrier proteins. So again, the channel is just like the little tunnel here. The carrier is the one that changes shape when a molecule gets to it, and then it carries it across the protein and lets it go. But these are all going from high concentration to low concentration. So these do not need energy. This one is going the opposite. It's going against the concentration gradient. It's going from low to high. And so if it's going against this concentration gradient, it is going to require energy, right? Remember, if it requires energy, it is known as active transport. And it's because it's going from low concentration to high concentration. Right. So again, just with things we talked about, passive transport, no energy is required because molecules are moving from an area of high concentration to low concentration. So it's like going downhill or going with the current. Examples are osmosis, which has to do water going from high to low concentration, diffusion, which is any particle going from high concentration to low concentration, and facilitated diffusion, meaning it's going from high to low, but it needs a protein to help it. Then you have active transport. Remember, active means that it requires energy, and ATP, right? Energy in the form of ATP. And so examples of this are a membrane pump that needs energy to pump things against a gradient. So it's going from low to high concentration. Molecules move again from that low concentration to a high concentration. And examples of this are endocytosis, exocytosis. And we didn't talk about penocytosis, but penocytosis is basically taking in liquids, whereas endocytosis is solids and exocytosis is releasing waste.